helps if you turn it on. Good morning. Thanks everyone for joining us. I know we'll probably have a few more folks that will be coming in a few minutes, but for those that don't know me, I'm Dennis Hancock. I get to be the Center Director for the U.S. Dairy Forage Research Center, which is part of USDA's Agricultural Research Service. And really excited to continue to bring these seminar series to you. This is the last day of the event, but we have these throughout the last 3 days of each expo week, bringing 6 different speakers from around the country to bring their latest research to us. Real quick about the center, if you want, after the program, you can check us out there at the booth behind the wall here. But our center is based here in Madison. We have 2 locations on campus in Madison, and then we have a 3rd location up in Marshfield. And I jokingly talk about the fact that we cover everything from crop to plop. So everything from soil and forage management through the animal, through looking at the microbes within the animal, and then looking at management of the nutrients after we put them back on the field. But it's my pleasure today to introduce a colleague of ours from a sister unit over in Pennsylvania. Dr. Kathy Soder is a specialist over there, a scientist that's been working at University Park for 25 years. I wasn't going to put too much of a spot on you, but a tremendous body of work that she's done over there, and I'm not just saying that because she's here. She's really one of my scientific heroes. I really appreciate her work. And I'm really excited for her to bring to you some selections of her work from Pennsylvania. So with no further ado, Kathy, I'll turn it over to you.
Thank you, Dennis. Thank you very much. Welcome, everybody. I'm going to discuss a few projects that we've had in interseeding and Brix for sugars and forages, and also some methane research in dairy cows, not necessarily our work, but our work plus some others. But before I start, I just want to give you a little bit of background like Dennis did on our unit. We are the USDA-ARS-Pasture Systems and Watershed Management Research Unit, and we work on supporting environmental and economically sustainable practices for farmers. We kind of do it interdisciplinary. I'm the animal scientist there. Started to do a lot of, I call myself a pseudo-agronomist because we're doing a lot of integrated work with crops, forages, and pastures and animals. And so we have the integrated project, we have a water quality project, and then we also have a modeling or a systems project. We currently have 10 scientists. You can see the variety of expertise we have there, from ag engineers to a hydrologist, soil scientist, ecologist. And then we also have a subunit in Massachusetts that does cranberry research. So that's a long story, but they are also part of our unit. So with that, I just want to go talk a little bit about the overview of some, like as I said, the methane research with grazing dairy cows, using Brix as a proxy for forage sugar or energy content in our forages, and also a long-term project we have going on with interseeding annual forages into corn for cover crop and for grazing. So the first part of the study is going to be a summary of an invited presentation I gave at the American Dairy Science Association conference in 2022, just summarizing some of that data. But when we look at enteric methane, the methane that's coming out the front end of our cows, you can look at different numbers and different sources, and you're going to see different numbers of exactly how much is being attributed to our animals, whether it be the enteric methane, the manure, or in the field. But livestock, if you look at the FAO data, produce about 14.5% of global anthropogenic emissions of greenhouse gases, and methane is the primary source. And it's been proposed that almost half, 40%, 47% of methane emissions are from grazing ruminants. Again, you need to look at different studies to see, and different sources to see what they're saying. But these grazing animals may have greater methane emissions because of decreased performance on high forage diets. And when we go to that, you have to look, when you look at a study, you really have to look at how they're reporting methane. Are they reporting methane production? How many grams per day is that animal producing? Are they reporting methane yield? How many grams of methane per kilograms of intake? Or are they reporting methane intensity? Grams per kilogram of milk, or energy-corrected milk, or some other production metric. It could be average daily gain if it was in beef animals. So you can have a lot of varying results, and I'm going to show you some of that data that's out there. But when we're talking about grazing animals, we know the research is much more complex, whether we're trying to quantify intake or methane. If we have animals in confinement, we can put them in a chamber, right, and measure the methane. We have a lot of options there on the way to do that. But when we have that grazing animal, it's a lot more complicated because they're not in a confined unit, they're moving around. There are other animals that may pull equipment off of them. And this is one of the older systems that's used with an SF6 gas, which is a carcinogen. But it is used. You put a bolus in the rumen of the animal with this gas in it, and you have to measure the release rate. There's a constant release rate, and it comes up out with the burps or eructations of the cattle. And we have an evacuated cylinder on the back of the animal here that's connected to a wire to the nose. And so if we have this constant of the gas and we know how much methane is in each burp, we can backtrack to an approximation. It's a very laborious process. We have done it. There's a lot of room for error. There's a lot of room for losing the equipment. So there's been some more advanced methods, and this is what's used primarily across the industry. I know there's quite a number of these green feed units at the Dairy Forage Research Center. And basically it's a head chamber unit. The cow, there's a bait, they call it, or a sample of a supplement. When the cow who has a transponder or an ear tag on her puts her head in this unit, it releases a little bit of feed, and that's what brings the animals to the unit. But every time the animal does that, there are sensors, and it sniffs. It sniffs the gas or the eructations or the breath of the cow. And if that cow's coming back multiple times per day, different times a day, we can get 24-hour diurnal variation in these animals, and we can collect data from a whole lot more animals. Again, not a perfect system, but again, it's more of a real-time measurement and a lot more accurate and easier to use than the old system. But if we look at our animal responses and grazing systems, if we're feeding animals in confinement, there's a lot more things that are consistent. We can keep the environment a lot more consistent, the temperature, even the humidity at times, or we can keep the feed constant in front of the animal because we're feeding out of silage bags or stored feed. It's pretty constant until you hit the next field or the next harvest. But with grazing animals, we have a lot of environmental changes day to day, even hour to hour, with heat and sunlight and humidity and flies and you name it. And forage quality and quantity, if we're moving these animals, that's going to change. Our management, they're doing a lot of forage selection, and I could talk diet selection all day because behavior is one of my favorite topics to research. And so there's a lot of that. How can we control that in these grazing animals, and how can we manage it? And then there's nutrient cycling. If they're gathering up around the water tank or a tree, they're depositing a lot of nutrients there, and it's not evenly spread across the field. So this does impact both our forage and our animal productivity. And there's always social interactions. If we have different animals, we have the boss cow, we have the submissive cows, and that's going to affect a lot of our consumptive behavior, especially in grazing systems where there can be competition for feed. And then there's individual animal preferences, previous experience. There's more recent indications there's epigenetic stimuli, what happens in the womb before the animal's born that can affect the animal's production later in life. But one thing we can help control is pasture management. If we get into more intensive grazing, and there's a whole lot of fad terms out there, whatever you're doing, something other than continuous grazing where the animal's on the same paddock all the time, we can help mitigate methane because we're getting a more efficient use of the grazed forage crops, and we're getting a more efficient conversion into meat or milk. So there have been some studies that shown a 22% reduction in projected methane annual emissions from beef cattle, and some of this data is in beef cattle, so I do include it. And so we can look at that by improving our management on pasture to help reduce methane emissions. Stocking rate of heifers showed no difference. If you had more animals, less animals, yes, there's a lot of contradictory information as well. If we have grasses with high soluble sugar, high carbohydrates, modeling suggests that the greater sugar content should increase methane. Greater digestibility, possibly greater intake, and intake is very closely tied to methane emissions. But another study showed that there was no effect of the high sugar grasses on dairy cows on methane emissions. So this is a very limited data set. It's very contradictory. So we don't have concrete recommendations that we can put out to reduce methane emissions in grazing dairy cattle. So some of the strategies we do have, and again, I'm abbreviating this, is animal management. And part of that in grazing is our grazing management. Genetic selection. There's a good bit of information going on genetic selection of animals that are higher, more efficient. The microbiome, that's another big area. That's a big unknown. We have this 50-gallon drum inside the animal that's this black box. It does whatever it wants with the feed and puts out whatever output those microbes want. So there's a lot of work on that. And the nutritional modulation. Can we feed supplements? Can we feed something to help reduce methane emissions? So we're looking for practices that promote more efficient systems that increase animal performance or fewer inputs that reduce methane intensity or methane emissions. So there's a number of feed additives, and you've probably heard of some of these. The 3-NOP is one that's they're working on coming out commercially. It has been shown to decrease methane emissions by up to 40% in dairy cows with little change in milk udder composition. And that's important because a lot of times when we're reducing methane, we're reducing production, and that's not what we want. But the results were affected by the fiber content of the diet, and I think there needs to be more work done there to tease out some of those differences. There's a lot of plant-based products, condensed tannins, saponins, essential oils. There's been work in oregano. There's been work in a lot of different plant-based products that show promise in vitro, in a lab, in a test tube, or in a fermenter system. But translating it to the animals is a lot more difficult because they are seeing a lot of reduced ruminal fermentation and productivity when they're feeding some of these products. Another one that I'm going to talk about a little bit because we have done some work in the seaweeds. That's kind of a hot topic right now, but there's some issues with that as well. So you can reduce methane by up to 98%. I'm going to show you some of our data here. But again, this was in vitro work, so it was small lab type work where we do it in fermenters. But what happens with in vivo when we put it in a cow? Does it translate into that? What's the cost of it? What's the environmental sustainability of that? And then there's a problem with the stability of the bromoform, which is that compound that is shown to decrease methane emissions. And if the bromoform isn't stable over time, it will decrease effectiveness. So again, very limited, if any, animal research data. So again, there's been a lot of work in beef cattle in relative feed efficiency, right? Some in dairy cattle, but really high in beef cattle. But we're getting much more interested in that from the environmental standpoint with enteric methane emissions. So it's very divergent. If you have animals that are more feed efficient, they can produce 20% less methane on a high concentrate diet. Another study showed that 20% less methane on a high quality pasture versus something that's maybe more mature or ranker than we'd like. And you can also decrease methane 12.5% in this study on grass silage versus a dry hay. So we can look at animals that are more efficient, meaning they're either consuming less and producing the same amount of milk, or they're just converting these nutrients more efficiently into our products. If we kind of focus in on those concentration strategies, there's a lot of contradictory information there as well. You can have increased ruminal fermentation on forage diets. That's fairly well established. And we can have increased ruminal passage and decreased ruminal pH as well with concentrate. Feed more grain, we're going to drop the rumen pH, right? We know that. So we can also increase our total dry matter intake by substitution rate. And that's really important on pasture or when you're substituting concentrates or forages because it's not a one-to-one substitution. If you get you can decrease forage intake by half a pound by increasing concentrate or grains by a pound, but you get increased total dry matter intake. So what does that do to methane? It depends if you're talking about production, how much you're producing per day, or intensity, because if we're feeding a little bit more grain, we should be producing more milk, right, if we have a balanced diet. So we're going to dilute the same amount of methane over more milk, so if somebody reports methane intensity, it's going to look more beneficial. We can also feed lipids to increase the energy density of the diet and decrease methane by 60% in some studies. But again, we have trouble with fats. You can only feed fats to a certain level. They're going to coat the microorganisms, might reduce rumen fermentation. So again, not an answer for everybody. So even though high forage diets have been shown to usually increase methane output, concentrate supplementation doesn't always decrease methane. A study, and some of these studies were conducted in Ireland and some other places that have been doing some really intensive work, and they're going to have different forages than we have here. There's going to be a lot of perennial ryegrass, white clover pastures. We have a lot more diverse pastures, a lot of different species than they do. But in cows grazing annual ryegrass and oat that were supplemented with corn silage or ground corn, the yield decreased. So the grams per kilogram of dry matter intake. So they were making less methane per pound or kilogram of intake, but the production, so the grams per day, they're still putting in as many grams per day into the environment, was the same, and the intensity, the grams per kilogram of milk, did not change. So, okay, we can say, oh great, we're decreasing it. Yield, yeah, because they're probably eating a little bit more, but they're not changing how much total they're putting out, so we're not really changing what's going into the environment. So again, if we take a look at this study, I hope you can see that just showing some of that. There was no difference in production. You can see, and the numbers, the absolute numbers aren't important. It's just the relative differences. But the yield decreased by about 6 or 8% when we did it. But you can see the difference in numbers just in how it's reported. And that's my criticism, a lot of the methane work, is we don't have a standard on how we report this data or even how it's calculated. And so we have a lot of questions that we need to answer yet as far as how we manage and control and respond to environmental concerns about methane emissions. So if we have a study here where they fed incremental concentrate feeding to dairy cows, and this is still in, so you're talking about 4 kilograms, and you can just about double those, 4, 8, 12, and 15 or 16 pounds of supplement to the cows, and these were grazing dairy cows. And if we take a look at the results, the production and the yield were similar, so the grams per day did not change. But the intensity decreased. The grams per kilogram of milk produced or energy corrected milk were diluting the same amount of methane over more pounds of milk. So, did we decrease methane? Not really, but if you report those lower numbers, it looks like we did. But they reported in this study that they counterbalanced it by decreased fiber intake because they had a concentrate substituting for a fiber, and no net change in methane emissions. One thing that's really important to consider too is they did see a milk increase with the increased concentrate. Obviously, we should see that. But we really need to look at the bigger picture, the life cycle analysis. There's more to it than just that methane, especially in pasture systems. We might be putting carbon into the air, but what are we sequestering in the ground? What are we doing for nutrient cycling? And those are really tough questions to answer because they take a long time, and they take a lot of different disciplines in order to do that. So I want to just mention one of the studies that we looked at a number of years ago where we were feeding flaxseed, which is an oil seed that has been shown to decrease methane emissions in cattle. And we did a couple different studies. This is the continuous culture in vitro study. We also did an on-farm study, and we did a controlled study up at University of New Hampshire. So we had orchard grass diet in this in vitro study, supplemented with 3 different levels of flaxseed. There was no flaxseed or control, 5, 10, or 15% of total intake in flaxseed. And when we looked at some previous work in cattle, it looked like about 5, 6, 7% was optimal for production. But in these systems, it's nice because we can go over the top and push it and see what happens. We aren't going to injure or have an adverse effect on animals in these fermenter systems because it's a laboratory method. And you can see that as we increased flaxseed, the methane production, grams per day, did decrease. So we did have an effect, but you can also see that about somewhere between here, it leveled off because between 10 and 15% there was no difference. So somewhere between 5 and 10% from a methane standpoint seemed to be ideal. But nutrient digestibility decreased. We have organic matter digestibility in the red and neutral detergent fiber in the blue. And you can see as we increased flaxseed, we decreased digestibility. How's that going to translate into the animal production? We can all guess. I mean, we obviously can't tell from this study, but we can guess what happened there. So when we went to the in vivo study at University of New Hampshire, we kind of saw the same thing. We fed it 6% flaxseed, and we did see a decrease in milk production and decrease in some digestibility. So even though it does decrease methane, The bottom line is flaxseed is expensive, it can be difficult to source, and it may impact your milk production. Another hot topic are the seaweeds. And we've done some work with the red seaweeds, the Asparagopsis taxiformis. And it's an aquatic plant that's native to Portugal and Australia. And we did get it imported from Portugal on a small scale because we were doing another in vitro study. And it contains, I mentioned, the bromoform, which inhibits certain types of bacteria. So it's hitting some of those methanogenic bacteria and decreasing them. So this type of seaweed contains very high levels of bromoform and other compounds that seem to have that effect. So again, we had another in vitro study, and it's kind of, these are our fermenters. They're 2-liter vessels that we inoculate with rumen fluid from cows, and then we can basically feed them and measure the outputs, the volatile fatty acids, the pH, the ammonia, the digestibility. And it makes a nice screening tool to then take to a cow study. So what happened with this study, when we fed the 0, half, 1, and 1.5% protein, or seaweed, you can see the methane basically went to 0. We did it and we redid it and said we are not registering anything. And so we're really looking at that. We think this was an artifact of the data. Some of the grams per day, there was some leakage in the one system, so we think that was an artifact. But we do 4 or 5 different runs of that, and every time it dropped it to 0. But we also got a hit in nutrient digestibility. We have dry matter, organic matter, and neutral detergent fiber digestibility, and you can see the trend, a linear decrease as we increase the level of red seaweed. So it probably is not an ideal forage or supplement. And we also, it's expensive. This was wild caught or wild harvested, very expensive to do. The amount of it's very small. So can we get it commercially? Can we afford to have it commercially? Are they going to farm it? And what's the environmental impact of that? And the other problem we found was that the bromoform degraded over time, because we had a couple different studies we used the same seaweed with, and the bromoform was less than half of what it was when we started. It's very sunlight sensitive, so we had to keep it in the dark. So sure, it decreases methane, but at what cost? Another forage that has been shown to decrease methane is forage brassicas, and we had some work done with that. I had a postdoc who was also in Dennis's lab at one point, and she did a lot of work with forage brassicas, because we can plant these in our similar climates to have either a summer slump feeding or a late fall feeding forages to extend the grazing season, because you have high biomass, high forage quality. And in vitro, again, because we do a lot of this in vitro work to start with, there was a reduction in methane emissions. And so we can use this as a cover crop or for grazing to extend the season. But how much of an emission effect is it having? We've done some of this in vitro, but are we really affecting it, or are we affecting productivity? And again, a lot of this needs long-term studies. If you do a 6-week study, what's it telling you in long-term on the cow? If you feed something to the cows, and let's say we feed it for all season, these are a short season, but let's say we have something else we feed all season, do they lose body condition? Do they rebreed? We don't know those effects on all of these things. But the forage brassicas do have a benefit from a grazing standpoint, because we can use them to extend the grazing season, produce some high-quality forage when we don't have it otherwise, and possibly decrease methane emissions as well. So as part of that, at the University of New Hampshire, we conducted a forage canola study, which is a brassica. And we had a controlled treatment where the cows were in the barn on a baleage the whole time, or our canola treatment is where they were in confinement in the baleage during the day. They came in after the morning milking, were in the barn all day, and then went out on a canola pasture at night. It's because you don't want to feed 100% brassica for rumen fermentation effects. And it was a 5-week study, and that is a criticism. But again, this is kind of the time frame you would have these brassicas available. And when you have animals available and budgets and everything, you do what you can. So we had a 5-week study, and 2/3 of the baleage was replaced by that canola at night. So all we did was reduce the baleage in, they were getting some other things, a little bit of grain and some minerals in a mix, but we reduced the baleage by 2/3. So the forage canola did increase dry matter intake by 5%, but milk production decreased by 5 to 7%. And it did reduce, feeding the canola did reduce methane production, yield, and intensity, so all three by 15%. But those trade-offs must be evaluated. You know, what's your bottom line? If you're extending the grazing season and saving forage costs for your stored forages, great. You know, if you're really looking at your milk yield and it's decreasing, you may not, and you may try different levels of it as well. You may have less of it. You may reduce the baleage by half or a third instead of 2/3. But that does show that there is an effect of these brassicas on methane emissions. Another thing that can really help is inclusion of multi-species pastures. You know, we talk a lot about biodiversity in pastures, and there are benefits on the primary animal or plant productivity, the secondary or animal productivity, overall greater animal nutrition. They can select, they can get different nutrients from different plants, greater drought tolerance. There's all kinds of reasons that we want to do mixed species pastures. So some of these studies, these were Irish studies where they did have some biodiverse pastures, not just their ryegrass and clover. So they were fed a diet with high legume proportions, and they were about 30–40% legume, and they did see a 20% reduction in methane emissions. So they had this white clover inclusion because it did increase ruminal passage rate, and anybody that's fed a high legume forage, you know, you don't stand too close behind the animals because it's coming through pretty quick. That's the passage rate. And the white clover could potentially impact the methane emissions. It could increase your milk yield or your solids, but with the same or lower level of methane, so you could increase your efficiency. Same methane, more milk. And they showed they have some results on some pasture herbs, we call them forbs, that have been shown to contain high levels of bioactive compounds. I talked about the tannins and some others. They had a mix of sorrel, daisy, yarrow, knapweed, plantain, and they fed it as haylage. And they did see a 10% lower methane yield than when perennial ryegrass pasture. So they saw, you know, looking at these, there's just potential for greenhouse gas abatement with a legume inclusion or a more biodiverse pasture. But this is very preliminary work and would really need to be done in a lot of different climates, because our climate's going to be very different from theirs as far as our forage composition. The other thing the white clover did, and I'm not going to get too deep in the weeds in this because it's not my area of expertise, is that rumen microbe population. And again, this was the Irish study at Chagas, and they looked at the sword type and how it altered the microbial ecosystem in dairy cows. And so they had white clover inclusion, and they saw a 12% decrease in methane yield with no effect on milk yield. And you can see what happened here. This is a simple graph to show how you had shifts in the microbial populations. And this was a lot of the methanogens, but some other ones as well. But they had a whole ton of graphs, and I didn't want to get lost in the weeds on these. But we can shift the microbiome through genetics, but we can also shift it through what we feed. And maybe we can do some work there on doing something more naturally, or selecting for animals that naturally have these lower methanogens, because there are differences from animal to animal in their microbiome makeup. So that was pretty much a summary there. So keys to reducing enteric methane in grazing systems. Well, the best thing we can do is make our animals more efficient. Let's get them calved out, let's get them rebred, right? If we can do that, we're going to be more efficient all around, and that will help because you will need fewer replacements, you'll have less downtime with your dry cows, and where we're producing more, because they're going to produce methane every day, right? So if we're not getting anything out of them, it's kind of dead weight, so let's keep them productive. We can improve our milk production per cow. I mean, I'm not saying maximize it, especially in our grazing systems, but improving it. There are trade-offs. You know, you're going to produce more methane, you might have more issues rebreeding, you got to have enough energy in the grazing forages to do that. You may need to supplement. We can certainly do improvements in our grazing management. I don't care how good of a manager you are, there's always somewhere we can improve. We can improve our forage quality and possibly our diversity in our pastures. We can consider supplements and feed additives. Again, you know, I mentioned a few, and there's some out there, but you know, keep watching the research, keep watching the trade journals. You may see some that come along that may be very efficient and practical for us. And then improving feed efficiency through genetics, nutrition, and management. And I mentioned life cycle assessments a little bit ago, and I just wanted to mention that because we really need to evaluate that integrated plant, soil, animal ecosystem, right? We can't just take one or the other. They all interact with each other. So we have to look at all these inputs and outputs to be considered. But that is complex research. There's no one person that can tackle that because it takes a lot. If we're looking at the crop, if we're looking at inputs being imported, we import a lot of grain from the Midwest. We're looking at fossil fuels. We're looking at a lot of things versus maybe improving our forage quality. So we need to look at our outputs, our meat, milk, manure, live animals, and then the fossil fuels used on the farm. So we're trying to integrate and do these life cycle analyses to get greater biological efficiency in our systems all around for farm profitability and environmental sustainability. And just a graphic, that's a simple one. We could really get complex on all the inputs and outputs on a farm. So the methane summary is, in general, grazing ruminants will produce more methane per day, the grams per day, and sometimes per unit of output to confinement systems. But it's only part of that carbon footprint. It's only part of that life cycle assessment. What are we putting into the ground by having these permanent sods? We're sequestering a lot of carbon. We're doing a lot of good things that I think need to be taken into account as well. And we need to look at all those external inputs. And I always say, it's not a we versus them. It's not like, this system's better than that system. We can all improve. We're all needed, and there's a place for all of us. So I think, we just need to keep improving everybody with everything and all of our systems. So with that, I'm going to switch to a couple of studies that we have going on right now. This one was one that has really come up with farmers and some consultants looking at using Brix as a proxy for forage sugar or energy. And if anybody's not familiar with Brix, I'm going to explain it in a minute. We just put this publication out, this manuscript out this year. So if we take a look at our forage plants, right, through the day, if we have growing plants, they are going to accumulate sugars during the day in the sunshine, and they're going to use them at night. Right? And there's been a lot of work. Hank Palin did a lot of work in Idaho showing that cut your hay in the afternoon, more sugar's in it, things like that, which is great. So the sugar contents are going to be higher, and they're going to vary. We have the months of the year down here, and then PM's in the light blue and AM's in the dark blue, so you can see where the sugar, how it varies through the year and through the day. So if we have increased plant sugars, we're going to have greater energy available for utilizing that high protein that's in our pasture forages. So we can capture more of that protein because it takes energy to do that, maybe get more efficient animals, get more productivity. We can increase dry matter intake and, as I mentioned, production. But these sugars are impacted by a lot of factors. Our forage species, our maturity, sunlight, temperature, day length, soil fertility, there's a lot of things that can affect it. If you have dew on the leaf when you test it, if you're testing it in the field. So the results may not be consistent. So as I mentioned, we've had some farms and consultants that have been using and promoting Brix to estimate sugar content. But what we have to remember is they're saying Brix is a measure of the sugar in solution. It's dissolved solids, so it could be anything. And this Brix is used in a lot of industries like the grape and wine industry. It's used in salt concentrations in saltwater aquariums, immunoglobulins in colostrum, and it's also used in the beer industry. So all Brix is you use a refractometer. You're extracting the liquid out of the forage or collecting it from your fruit or whatever your industry is, and it's just measuring the angle at which it reflects. And again, the dissolved solids is the important point here after a bit. Because our forages contain a lot of things other than sugars, oils, minerals, pectins, acids, fiber particles. There's a lot of things that can be in there that may be causing a reflectance and really isn't a sugar content. So is it a reliable measure of forage sugar? So that's what we want to look at. We took some forages. We took alfalfa and orchard grass established fields, and we measured them 4 times through the season, and we compared them to wet chemistry analysis of individual and total sugars and nutritive analysis. So 2019 and 2021 is when we did this in central Pennsylvania on 2 well-established fields. We made sure we had a sunny day, a warm day between 75 and 80 degrees Fahrenheit. The dew was off of there was no rain or dew on the forages, that they were dry. And we just crushed them in a garlic press and then read them on, we had this is a simple refractometer you can see in the field. We had a refractometer that was more lab appropriate, so we used that, but we just pressed the forage out, or pressed the juice out of the forage and read it. So then we took forage samples and dropped them in liquid nitrogen for nutrient analysis so that the plant didn't keep respiring and change the sugar contents. So what we found here is we looked at total sugars, and then we looked at some of the individual sugars, the fructose, glucose, and sucrose, and we have alfalfa on furthest from me and orchard grass closest to me. And when we looked at fructose and glucose, no difference in the harvest. So we had harvests that were roughly June, July, August, and September for each of the forages. We did it about once a month. No difference in fructose and glucose across harvests. Sucrose was highest in harvest 4 for the alfalfa and highest in harvest 1 and 4 in June and September for orchard grass, but it was lowest in August for us, just for the orchard grass, but not the alfalfa. Total sugars were highest in harvest 1 and 4, June and September. That's when the plants are growing rapidly. You expect them to have more sugars at that point. When we looked at forage quality, there were some differences. Highest in September for the alfalfa, highest in June for the orchard grass. So we're seeing some differences between that legume and that grass. And then when we look at the neutral detergent fiber, or that was protein, sorry. When we looked at neutral detergent fiber, it was highest in June in alfalfa, lowest in September. And for the orchard grass, it was highest in August and September. So we had higher fiber at that point. And then we compared the Brix index. Throughout the season there in the alfalfa, there were no differences in the Brix reading, but in the orchard grass, it was lowest in June. The other 3 harvests were not different. Here's the interesting data, and this is what I want you to take away. We did correlations. So if we had a perfect correlation, when Brix went up, sugar would go up. When Brix went down, sugar would go down, because that's what the indication is. So we have a no correlation is on this line at 0. A perfect correlation, positive correlation would be here. A perfect negative correlation, meaning if Brix went up, the nutrient or the sugar went down, which is opposite of what we want. And you can see they're all over the board. In harvest 1, probably with lower fiber in June, we did a pretty good job of estimating sugars with the Brix in alfalfa. Okay, but you can see this one. So this tells me that when the Brix reading was higher, the sugar content was actually lower when we did wet chemistry. But look at the other harvests. It was actually opposite of what we wanted it to be. So it's not doing especially in the grass. The grass was worse than the alfalfa. So it wasn't doing a very good job. So then we looked at it from nutritional quality, just for fun. People aren't really predicting protein or fiber out of it, but when we looked at that, because we have protein in blue, acid detergent fiber, nitrogen detergent fiber, and hemicellulose. So we're doing 3 different components of fiber. For the most part, which we would expect, as the fiber got higher, the Brix was lower. But we had some of these other data that it just wasn't doing a really great job of predicting it. So it may not be accurate enough to quantify sugars and forages to say, oh, the sugar's higher in this pasture, I'm going to put them in here right now, and it's higher over here tomorrow, I'm going to put it in here. Because our sugars are much lower in concentration than what's in grapes or in some of these other industries. We only have about a quarter or a third of what they are, so it might need more sugars to get an accurate result. Plus we have all those other items in the solution. We've got those pectins and those tannins and different things in there that might be causing some noise for the refractometer. So Brix was developed for fruit and food crops that are largely comprised of sucrose and fructose, while forages are comprised more of fiber and many other cell solubles. We don't just rely on the sucrose for energy out of the food, we also get it out of the fiber component as well. So based on this study, Brix measurements do not directly correlate to forage quality, especially energy content. I do have a fact sheet if you didn't get one when you came in on this study. We're really putting that out, and just, farmers really need to look at other ways of estimating, because Brix is so affected by different factors: time of day, if there's a drought or a wet year, temperature, humidity, dew, human error, how we squeeze it. We found if we change person to person, we got different results. So it should not be used as a sole variable to estimate forage quality. And farmers may want to look at other methods to be more accurate testing. We're getting field level NIRs out right now. They're coming on board. That's probably going to be a much better option than this. A little bit of an investment, but still more accurate and may help people improve how they're feeding their animals on pasture. So the last thing I want to talk about is a long-term study we have going on right now looking at integrating livestock and cropping systems. We're interseeding cereal rye into standing corn for late season grazing. So if you look before World War II, farms were very diversified. You didn't have monocultures, you had a lot of nutrients returned to the soil, you didn't have imports and exports off of the farm, but then we had the Industrial Revolution, we had the war, and all at once nutrients and everything are moving all over. And farms became much more specialized. They're growing monocultures, whether it's corn and soybeans, whether it's other crops, and they only have maybe one industry on their farm. Maybe they're strictly a dairy or strictly something. They lost all that other diverse income that they had. And we've had a lot of negative consequences because of that. Our water quality and groundwater supplies, erosion, nitrogen, loss of nutrients and eutrophication. I'm in the Chesapeake Bay, and that's huge right now. We're really limited on nutrient application because of that. Increased insect and weed issues, if we're getting out of our biodiversity. So we want to look more at integrating crops and livestock because we do have a growing human population and we have greater pressure to produce more food on the same or probably shrinking land base. So we're looking at combining these enterprises. Can we grow a crop and graze animals on it? Can we grow a crop and do something else on it? But we also want to keep in mind the environmental cost or benefit. So when we look at grazing our annual forages, we're bridging that gap in cool season perennial. That's our typical cool season perennial growth, right? We got a big flush in the spring, it kind of dies off in the summertime, and this time of year it's growing again. But we have these gaps. We've got gaps on both ends, and we've got the gap in the middle of summer. We can use warm season annuals to fill in those gaps. We can plant some sorghum Sudan, we can plant some different things to put in there, we can have some small grains planted to hit that late spring season for grazing. But we can also plant some other things looking at cool season annuals. I talked about brassicas before, and I've talked about there's other forages out there to help even out that forage supply throughout the year. So we're trying to reduce our stored forage costs and needs, and also maybe utilize more forage that we already have out for crops. So our premise was that corn harvested as grain is harvested too late to get a cover crop established, and it certainly would be the issue here as well. And this uncovered soil has all kinds of problems, right? We like to keep the soil covered. So these cover crops that we put on can provide conservation benefits, but can also be a low-cost source of livestock feed. And so we were looking at the trade-off between crop productivity, that corn crop, if we graze or don't graze that cover crop, and these environmental effects are really not well understood. Work that was done at Penn State by Bill Curran and Greg Roth a number of years ago looked at corn yields if they had no cover crop, red clover, annual ryegrass, or a combination of red clover and annual ryegrass. And you can see that there was a distinct effect of these cover crops on corn yield, providing nutrients, providing maybe some soil cover, maybe keeping the soil temperature down or keeping more moisture in the soil. And the combination was the best, but when we're starting a study, we like to walk before we can run. If we put too many variables in there, we don't know what the cause is. So again, we looked at evaluating long-term effect of interseeding cereal rye into growing corn for late fall and early spring. And we are currently in our 8th or 9th year of this study. I have 4 years summarized in this. So what we're doing, again, we have that graph of the cool season forages or perennial forages. So we're planting the corn, hopefully in mid-late May. It didn't get until June this year because of the weather. And we're interseeding the forage in about a month later when the corn's about V4, V5, about knee-high. We're going in so before it gets too tall that we're not knocking it over. So we're harvesting the corn in late October, early November, ideally, and then we're grazing it about 4 weeks later. And we do get growth after we harvest that corn. It's amazing how much growth we get. And then if it regrows in the spring, we're grazing it again before corn planting. And so we've been repeating this. It's been repeated corn. We have not rotated it out. We have 2 sites, 12-acre fields that we divided into 6 2-acre paddocks, 3 grazed, 3 not grazed on each site. And then we were using 95 to 98-day relative maturity corn. We have since gone to an 85-day corn so that we can get it off a little bit earlier. Planting at 26,000 plants per acre, applying some fertilizer, starter fertilizer. We're putting side dressing at interseeding. And we're using an interseeder that was developed at Penn State. And it goes between the rows of corn and puts in 2 or 3 rows of the annual forage. And again, side dressing at the same time. So again, the corn is harvested, and we're taking corn grain biomass yields, allowing that rye to grow, grazing it, and then taking some samples for biomass. And so what we found is we grazed it with 24 beef cows that were adapted to grazing corn. We found out that was important. And we moved them every couple of days, every 2 to 4 days. And these animals, we didn't take animal measurements because of the way it was set up. They were only used as harvesters. Spring grazing, we started at 8 to 10 inches, but that's kind of a debate right now on when we should start and how it grows, and because we didn't spring graze every year. And we did spray before replanting, and our non-grazed plots were mechanically harvested. Bottom line here is we saw no difference in corn yield between the grazed and the non-grazed. The grazed is the green, the non-grazed is that yellowish orange calf scour-like color. The only difference we saw was this year in 2019 because it was a drought year and the corn yields were down all over the place. If we look at our pre- and post-grazing corn stover and cereal rye, we had a dry year, of course, in 2019. We had really no differences within year between the two. The corn, the solids are the grazed paddocks, and the hatched are the non-grazed, and then you have your corn and your rye, so it give you the difference in the bars. Beforehand, not, but again, afterwards, we certainly reduced the forage amount in the biomass yield because we had the grazed in there. And we had about 24 to 26 days of grazing on the 3 years that we grazed. We did not get to graze in 19 because there wasn't enough forage. Similar in the spring, but the only year we grazed was 21. And this year, it was up over the cow's back. It was growing so fast. And we don't know what we did right that year because it's the only year it did that. So lessons learned, we used dairy heifers one year that were used to grazing, but they did not do well on it. We grazed too late. We used long season corn. I went too fast there. Yeah, we used long season corn. Again, we're using 85-day corn now. Spring grazing is not as consistent as fall. We probably need a higher stocking density in the spring when we do have spring grazing, because you can see where this was up to their noses whenever we were in there. And that was getting towards the end of it, but we could have used more cows. But we often had to pull the cows off too early because we didn't have enough cows because it was time to plant the corn. And to date, we have not decreased the corn yield as a result of the grazing or of the repeated corn plantings. So moving forward, we are looking at adding cattle, evaluating mixtures. We have a plot study now where we added brassicas into the rye, and we're looking at some different nutrient use efficiency. We have some different nitrogen application rates, soil compaction, and soil organic matter. So with that, I am going to quit. I can answer questions. I do have a fact sheet on the interseeding study as well if anybody's interested, if you didn't pick it up.
All right, thanks, Kathy. Very good coverage of a lot of great research that you got going on there. Any questions that we have from the audience? Unfortunately, I do need to pass the microphone to you. Go ahead,
Hi, Kathy. Thank you for the presentation. I'm very curious about the methane section of your presentation. You talked about a lot of studies that are more small scale, in vitro, in lab, and also larger scale, in vivo with animals out in the feed or on feedlots. I'm wondering if you could explain a little bit what's the importance of each one of those two types of experiments, what kind of questions each one of those will answer, what's the importance of having both?
Right. Well, as I mentioned earlier, a lot of our in vitro studies can be a screening. We can titrate levels, like feed those different levels and say, hey, this is the ideal one, let's take it to the cows, like we did with that University of New Hampshire. I didn't report that data, but we did that. So that's what we do there. Or does it work or doesn't it work? Or did it kill rumen fermentation? We might not want to feed it to cows, type of thing. There's all kinds of questions. So that's there. The shorter term stuff, again, can give us some data, but we don't have those long-term studies because those grain feed systems are probably 80,000, 90,000 a piece right now. You need so many of them. I haven't bought one lately. Mine was 70 something, so I'm sure they're up to that now. And so there's a lot of, and grazing research is just so labor intensive. It takes a lot of land, it takes a lot of labor, and we need it, but it's challenging. So.
Yeah, those are not cheap, and also they come with a very expensive yearly maintenance plan that goes along with it too.
And training too, because we went out and took.
As an administrator, I can I see those receipts
yes.
now. Other questions? Yes, sir.
Yeah, thank you, Kathy. That was a great presentation.
Thank you.
I've got a few questions. First, on the I'm looking at it from the greenhouse gas emission studies, using different strategies. I think the first question is on using fat.
Using what?
Fat.
Okay.
Fat. I mean, you presented on flaxseed, right? Which contains a lot of unsaturated fatty acids. So I was just wondering your thoughts on things like your high saturated fatty acid type of oil, like palmitic acid or stearic acid, whether you've done anything in that space.
I know there's studies out there. That's not my specific, I haven't been through that, but there are some on extracting some of those fats and feeding fats, but we also have a limit on how much fat we can feed. I should mention too, our flaxseed was whole flaxseed, not extracted flaxseed, so we had the whole fats in there. Because on some of the studies that are out there, you'll see they fed extruded flaxseed, which is a different level of, there's not as much fat in there. I think it's a possibility, but again, we're going to have to really evaluate ruminal fermentation, especially if we have animals on pasture who might be on the edge of some really lush pastures. Is that beneficial or is it not? Yeah. But again, I'm not totally familiar with that. I just mentioned it as a strategy, but it's not one that I've done.
Yeah, no,
other than
I was
flaxseed.
just wanting to hear your thoughts on that. Sorry, apologies. I've got a few other questions, especially in relation to concentrated feeding and different forages. You mentioned canola and other brassicas. What do you think is happening here? Is it because of changes in rumen fermentation profile from maybe more of acidic to propionic acid ratio, or is it just a matter of dilution effect on fiber content?
With a lot of the brassicas, you're seeing these secondary compounds, the main one being the glucosinolates.
Right.
And that is affecting ruminal fermentation. Again, when you get into the microbiome level, that's a fairly new area. And you want to talk about another expensive area to do analysis in. We've dabbled in that a little bit with the help of a microbiologist, because it's, she goes through all the rattles this stuff off. But it's a new area, and I think, with some of these forages, we might be able to get some extractions. That might be another example. Can we get the bromoform out of the seaweed? Can we get the glucosinolates out without having the negative effects? Because glucosinolates, a lot of these secondary compounds have negative effects too on intake or production or flavors in the milk. So we have to watch that. With the brassicas, you have to watch off flavors in the milk. And
I will leave everyone else to ask questions. I'll come back.
Please, if you want to go ahead, if you have more questions.
That's it? All right, the other thing I was going to ask you, apologies again, is about the seaweed. And there's been some thoughts on or concerns regarding heavy metals. Mm-hmm. What's your thoughts?
We did not analyze ours for heavy metal, because when we did that study, that really hadn't come to the forefront, and we didn't have the sample oil. They were too old to, I think they were gone by the time that came around. If there are heavy metals, it's certainly going to be a concern from especially a human health, because some of the other stuff, some of the seaweeds, the other thing that you have with them, with some of them, is iodine. And kelp, which is not used for methane suppression, but is used as a mineral source. If you get high kelp levels, and we've done some work with University in New Hampshire on that, they took samples of milk from the stores and from processors and from the farms, and there were some very high levels of iodine that could be a concern for children, that they may be toxic levels. So there's a lot of things we have to look at. Yeah. The heavy metals specifically, I don't have, but there are other things in there. Yeah.
So yeah. All right. Thank you.
And not to steal thunder from our speaker this afternoon, but Lizzie French, who's speaking at 1:30 this afternoon, has looked at iodine and some of the bromoforms in the milk, and she can really speak to that. But I think it's a negligible issue, not totally out of the question. But yeah. Go ahead.
Kathy, on the Brix, at the end of the Brix talk, you talked about it may not be reliable enough for farmers, and you started to mention something that they might be able to use. Can you expand that a little more?
The handheld NIR, near infrared spectroscopy is what it is, and it's typically a lab unit that uses light waves to analyze forages or feeds. And we've had them in a laboratory, and you can send them away and get NIR analysis on your forages at any of the commercial labs as well. Wet chemistry is always the gold standard, because we're getting this is an estimate using a lot of equations and light refractions and all of that. But there are developing some handheld units that may be able to be used at field side. And I don't know exactly. Are you familiar with where any of that is, or?
Yeah, actually, Probably one of the best ones here locally is actually some of the folks at Dairyland, our partners over with the Forage Analysis Super Bowl. They've been doing a little bit of work. I don't think Kyle is here today, but Kyle Taysom is one there that's done a lot of work. And also at UW, Matt Digman at UW-Madison has done a lot of work with the NIR samplers. So those would be some contacts there too. Other questions? Sure.
I guess at the end of the day, the question is, what do we do going forward? Is this something we really need to worry about, or is it focused on animal performance, efficiency, and is that what we should be thinking rather than all these abatements?
If a grazing dairy farmer, I was out on a farm yesterday, if a grazing dairy farmer asks what he can do relative to this, to methane in particular, get your forage quality up, get your grazing management up, and get rid of those inefficient animals. You know, that's going to go a long way.
Kathy, I'll ask one more, I guess. I interviewed a farmer earlier this summer who's actually got a grant looking at one of these energy grants looking at how to reduce methane in beef system. And he was talking about he couldn't get red seaweed, so he was feeding some other seaweeds. Is this really, think, viable from what you've seen so far to add the seaweeds?
Economically, no. Because of the limited source, you know, and then there's the cost of harvesting it wild. There's the economic or the environmental impact of harvesting it wild, and is there enough? And then if we go the other route and we farm it, there's a whole nother level of sustainability or not there as well. And so I think that's a lot of the questions right now, whether it's going to become an economically and environmentally sustainable way to do it. Just because we can doesn't mean we should. And I'm not saying yes or no, because what we showed, it's very effective, but you know, we had all these bigger questions. And then there's the question of, you got to keep it, at least what we have, keep it in the dark, and because it breaks down the bromoform, and then just bromoform stability in general. If they could find ways to stabilize it, or as I said to this gentleman, to extract it, and maybe just use that compound and develop it, there may be an option there too. You know, it's like we found aspirin effects in willow tree or willow bark. You know, now we have aspirin. You know, there's different things that.
And I think that's a really great point. I think one of the great black boxes of agriculture are the secondary metabolites and secondary plant compounds that are produced, and there are a lot of those that are very metabolically important, but we haven't really sussed out exactly how we could utilize them. So. Well, Kathy, we sincerely appreciate you coming down. I do have a couple of things to give you as a token of our appreciation for your travels. Fortunately for you, it's actually official because it has the USDA logo on it. A cup there for coffee and an igloo cooler there to keep everything cooled as well.
It works. Thank you.
So really appreciate Kathy doing this and coming down from Pennsylvania. But also, she'll be around for a little while if you have some questions. And let's give her a round of applause.
Thank you. Thanks for coming in for the good questions.
We do have one additional presentation today at 1:30. I encourage you to come back to hear Dr. Lizzie French talk about some of her work, particularly with automated systems in the dairy industry. So I'll see you there at 1:30. Thank you.